Hi everyone, I'm Tapushi Kormakar. Welcome to my channel Pure Biology. And I have already started the Lac Operon topic in the previous videos, and I today continue the mutations of the Lac Operon system. Okay. Before this topic, I already explained about the regulated gene mutation in my previous video, and another also topic related to the Lac Operon also I have described. So you obviously check these videos, and after that you follow this one. Okay, and I give my link on the I button of the video, and so from there you can check out the videos from there, or obviously you can check out that videos from the playlist. And before going to this topic, I also want to say thank you to all of you who have already subscribed my channel, and for the growth of my channel. Okay, and obviously I request you, if you will get, or you are getting the information from the from this channel, obviously that related topics and that are very important for the biological science. So you share this channel with your friends, and obviously subscribe my channel for the growth of this channel. Okay, so let's start. This is the topic Lac Operon, Lac Mutation, and today I just describe you about the Lac OC. C means constitutive. Okay, so in case of Lac OC, generally Lac O plus, that is normal. Okay, that is normal. Why it is normal? Because the regulator gene that produce the repressor proteins, and these repressor proteins bind to the operator system, and then in presence of inducer, this cannot bind continuously to the operator system, so the transcription is possible of these genes. And if inducer is absent, in that situation it binds to the operator system continuously or for the longer period of time, and the transcription is not possible. So what happened in the replacement, or instead of O plus, if the OC is there, that means the operator is not O plus, wild type, this becomes OC. Then what situation arises? OC generally is cis-acting. Okay, this is cis-acting and obviously dominant to O plus. It is very dominant gene. After mutation of the operator system, this is very dominant gene of the wild type operator. Clear? And does not allow the binding of the repressor. Okay, this is very dominant and cis-acting. It is dominant over O plus. O plus is normal or wild type. Generally, the regulator protein binds to the O plus. Okay, but if the mutation is occurring in the production of in the replacement of O plus, suppose OC is formed there. So what happens? This OC is cis-acting and more dominant as usually. It do not allow, or it does not allow the binding of the repressor. That means the regulator gene produce the repressor protein that cannot bind to the operator system. Okay, and base pair alteration make it unrecognizable to repressor. That means the repressor cannot recognize this site. Clear? Repressor actually recognize the O plus site, so it can easily bind to the O plus site. But in case of the formation of this mutation, the repressor cannot recognize this site. Why? Because here the base pair alteration occur. Okay, during the formation of this, the base pair alteration occur, and as usually repressor cannot recognize this site, so it cannot bind there. And as usually repressor cannot bind there, what situation arises? The constitutively expression of the structural gene is possible. Okay, transcription, translation is occurring continuously. So constitutively expression is possible here. Suppose this is one type of example. There O plus OC, Z minus Z plus, and Y plus Y minus. This is diploid. And what happened in this situation? As OC is present, you just learn it that as OC is present, so inducer is present or not present in the medium, the transcription is occurring constitutively. Clear? As OC is present, so O plus activity, its activity is not necessary because it is dominant. And if this is present, so repressor cannot bind this site. Okay, so it is dominant over this one. So in that situation, what happened? The constitutive transcription process is occurring. Okay, so here inducer present or not present, the expression of the gene or the gene's transcription translation process, that is constitutively occurring. Okay, so here in that situation, Z minus and Y minus, so beta galactosidase and the parmy is produced. Z minus, Z plus and Y minus. So in first situation, Z minus Y plus, and second one Z plus Y minus. So what happened? Beta galactosidase and the parmy is both produced. In case of Z plus, here the beta galacto constitutively it exists. Z plus is constitutively exists, but the Y plus, that means the production of the parmy that is under control of inducer. If inducer is present, so the formation of the parmy is occurring maximum, but if inducer is absent, so parmy production is not more. Okay, but Z plus, that means the galactosidase, it's constitutively expressed. Why? Because Z plus is situated just adjacent to the OC. You can see here the Y plus and Z minus. In case of O plus, there adjacent to the O plus there is the Z minus, but here adjacent to the OC or O constitutive, the Z plus is present. So beta galactosidase produced continuously, but the Y plus production or the parmy's production is under control. Here, and OC effects on the genes downstream from it generally affects on the downstream of the gene. But O plus, that structural gene adjacent to it, O plus its activity just depends or its activity affects on Z minus adjacent to it, but its effect totally downstream to the gene. Okay, downstream to the gene. Clear? So its effect or its activity affect on the adjacent of the gene, but its effect that is seen downstream of the gene. Okay, so Z plus Y minus both are affected by the presence of OC. Clear? So this is O constitutive or the OC mutation. Another last mutation, this is the Lac P promoter mutation, and we write this P Lac minus. Okay, this is obviously cis-acting and uninducible. Cis-acting and uninducible. This affects on all the structures. Obviously, if P is mutated, that means the RNA polymerase cannot bind the promoter site. So transcription of the structural gene is not possible. So in presence or the absence of inducer, okay, you just imagine if inducer is absent or inducer is present, the P is mutated or P is minus, Lac minus. In that situation, the RNA poly cannot bind here. So, if inducer present or inducer absent, so that is not affected. The structural genes transcription is not possible and translation also not possible. So, these are the mutations. I have totally explained in two parts all the mutations that are related to the Lac Operon. And if I in my next video I also explain about the positive control of Lac Operon. Okay, positive control of Lac Operon, and also if I get some times, then I also describe about the sums that are very important and you will get in the CSIR net. Obviously, I will discuss with you. Okay, if you understand about the mutations that are related to the Lac Operon, wild type and mutations, so you can easily solve this type of sums, Lac Operon sums easily. Okay, so you continue with me and my channel. I will give you this type of descriptions or this type of informations in different topics gradually. Okay, so thank you.